First of all, welcome everybody to the kickoff program today for the 2014-15 Timothy Johnson Medical Scholars Speaker Series. I always like to point out at the beginning, for those of you who don't know who Dr. Timothy Johnson was, he was a faculty member here with the initial founding of the Virginia Tech Fralin School of Medicine, who was very involved with the development of the curriculum, the basic sciences, and the research component. Who unfortunately became ill and died about a year ago, Rick, or so. And we named this program in his honor and like to remember him whenever we get together for the program. And I know Tim would have been, actually was, because he came to the first one, was very proud and excited to be part of it. And it's great to see this series going on. Having said that, today is also the first day of a little bit of experiment we've been running, and all the students out there know about it because you're it. And that is, in addition to having the first year medical class being involved with this program as part of your research education curriculum in the first year, we also have the first year PhD program students from the Translational Biology, Medicine, and Health Virginia Tech program. And as you all know, you're working together on various things. So we're really excited, both for our speaker, for ourselves, and for all of you, to have this opportunity to have you all here together. So hopefully this will be a great start to that, and I'm sure it will based on today's speaker. So without further ado, it's my pleasure to introduce today's guest, Dr. Les Biesecker from the National Institutes of Health. Dr. Biesecker did his undergraduate work at the University of California, Riverside, and then did his medical training. We found out we share the same alma mater at the University of Illinois. And from there, he went on and did residency training in pediatrics at the University of Wisconsin Hospitals. And from there, moved on to do a clinical fellowship, the section of pediatric genetics at the University of Michigan Hospital. And after that, he went on, or stayed on, I should say, to do a fellowship in pediatric genetics as well at the University of Michigan. He spent some time as a faculty member at St. Louis Children's Hospital and then at the Mott Children's Hospital in Ann Arbor, Michigan. In the early 90s, he was recruited to, or joined, I guess you recruited to NIH, that's fair to say. He was recruited to the National Institutes of Health, where he's been since then, and has had a, meteoric is a kind of a hackneyed phrase. He's had a tremendous career trajectory. I don't want to say meteoric. I think it's meteoric, though. Anyway, you just look at his work and his productivity and the impact he's had in the last couple decades or so, and it's really important and really impressive. He's when you work at NIH, there's a lot of things you have to do and a lot of things you can't do being a federal employee, and it looks like Les does all the things you can do and helps out and contributes to the community as well as NIH in many ways. He serves as the chief there of the branch for human genome research. He serves as a co-director of the Sequencing Center. He's been chief of the Genetic Disease Branch at the National Human Genome Research Institute now for several years. In addition, he serves on several national and international panels and advisory boards, and he serves on the editorial boards of a number of important journals, such as the American Journal of Medical Genetics, Genomic Medicine, BMC Medicine, and the American Journal of Human Genetics. Dr. Biesecker's work, as I think the students know from reading some of his papers, has spanned enhancing and increasing our understanding of a number of genetic disorders, some of them rare, some of them not so rare. He's been a proponent of, an advocate of, and an eloquent and articulate communicator of the importance and usefulness of using genetic analysis, exome sequencing, and has dealt with a variety of important issues, not only a whole host of disorders that we could go through that affect intellectual development and dysmorphology and hypogonadism and metabolic disorders and so forth, but also dealing with some of the thornier issues about things like variants and incidental findings using whole exome sequencing now, and the very issues for you medical students out there that you will be facing immediately, the decisions you'll have to make based on your understanding of that science and how to apply it. And so he not only is a leader in the science of it, but I think he's also a formulating figure in our thoughts about how to apply the science and the medicine as well. And we certainly had fun. I know I did, going around the rooms the other day, and I know the students did, going over a couple of his papers, one on these incidental findings and cancer mutations, and another on identifying the ACSF3 mutation that leads to the aciduria defects. So without further ado, please join me in welcoming our guest, Dr. Les Biesecker.
Thank you. Very nice. Thank you very much. That's a lovely introduction. I'm very happy to be here. I enjoyed my dinner last night. It was great, and my talks this morning were really stimulating and rewarding for me. I was just thinking, as you were giving that introduction, it's funny that we use the word meteoric, isn't it? Because meteors are things that sort of come down, flame out, and cause huge amounts of collateral damage. I've never seen one go that way, though. That's what's fun. And no, I like it. We all use it. It's funny how you think about some of these things we say, and actually it's not right. Well, that's it anyway. Thank you very much. I love coming here. All right, so what I want to talk about today. First of all, I must disclose, as I have last night and to some today, that I'm not sure I'm a scientist. I'm not sure I'm a researcher. Does anyone know what this is? What? Yeah, it's actually a hand axe. It's about this big. I think it's 1.2 million years old. A fantastic book for you all to have on your bedside table. It's called The History of the World in 100 Objects. Has anyone read this book? It's from the British Museum. Absolutely wonderful. And it's just, they go through their collection and they take these objects and they tell you the story of these objects and what this tells you. So 1.2 million years ago, somebody created that out of a piece of rock. Okay? So the person who did that, I would suggest, is much more creative than I will ever be as far as creating a technology that one can use to do things that you want to do. I'm envious of whoever did that, knowing that I will never create something as amazing and useful as that. Instead, I consider myself a person who exploits tools, exploits technology. I'm just a medic. My colleagues in the UK, they call them researchers and medics. They don't call them physicians or clinicians. I'm a medic. And that's a disparaging term, and I'm really comfortable with that. I'm a medic, and my job is to look around and see what tools are out there that I can use to answer questions that I think my patients are posing to me. Now, you do that, I did that, I practiced general pediatrics for a couple years, and you have an armamentarium of available tests and therapeutic interventions that you can use on patients, and that's your job, is to look at your patient and figure out which of those tools are appropriate for which person in which scenario. Clinical research is not that much different from that. My job is to look for sort of nascent or developing tools that are coming around the corner, and ask the question, could that tool be useful to answer a problem a patient needs the answer? And also, arguably, I am captive to the tools that are out there. Because again, I'm not going to create the tools myself. People develop tools, and I look at them, and to some degree, the questions that I ask of those tools are actually driven as much by the tool as they are by the problem of the patient. So I take a tool and I say, what useful clinical problem can I address with that tool if I applied it in a certain way? So what I want to talk to you about today is some of the work my group has done in clinical research of using tools, and obviously we're going to talk about genomics and sequencing, and how that might be generalizable to think about how one could use those tools in clinical care, and how this thing that we have lots of names for this: personalized medicine, predictive medicine, individualized medicine, what that might look like, how you could potentially do that, and what some of the implications are. Okay, all right, so I'm going to use clinical research, but basic research, it would be very, very similar. What's our paradigm? What we currently do is this. We develop a question, develop a problem by looking around us, and then those of you who are First-year graduate students are learning sort of what the lay of the land is as far as what science we know and don't know, what are questions that are open, questions that are closed. Gather information to allow us to formulate some hypothesis, some constrained question that one can answer. So in clinical research, we look at patients, some phenotype that we want to address, and we formulate a hypothesis that this biologic attribute explains this phenotype. That's how we do that. Basic science the same. Then we gather a bunch of patients, and we, in clinical research, exquisitely, carefully detail and dissect their phenotype so that we can develop a group of research subjects who have exactly the attributes we want them to have in order to feed them into this process of answering a question that we want to answer. Then we apply this assay, some assay to the patients, participants, to test the question, and then he will either answer the question or not, and we use those data to either conclude that we have answered the question or, in fact, more often than not, refine the question and then ask it again. We iterate on this process, and then use that to advance our knowledge. Okay. This is our sort of hypothesis testing clinical research paradigm. And the most important thing I want you to take away, and I've learned the hard way, I have to say this at the outset, is this is a good paradigm. It works. It should continue to be used because it does generate useful and generalizable knowledge. But I want to suggest to you that it's not the only way to do research and how to think about answering questions. Because if you use this paradigm, what you do is you get yourself into a certain situation. And that situation is that you end up doing what? Does anyone know what this is telling you? Quick, that's faster than most groups. Paint yourself into a corner. And that corner is that you have constrained discovery by your input assumptions. Because you have defined a hypothesis, you can either answer yes or no, and you can only answer questions that are within that domain of ignorance. And you cannot get out of it because of the formalized process that we use. Many of us do this. I do this kind of work myself, and again, it works. But Mark Twain said, whenever you find yourself on the side of the majority, it is time to pause and reflect. All right? And so the question would be, well, is this the only way we could do this, or could we do this a different way? Well, here's another way you could think about doing things. You could, instead of the hypothesis testing mode, you could gather your cohort of patients and then just generate huge amounts of data about them. In fact, you could even envision, as is now technically possible, you can generate all possible data of an attribute, which is kind of a radical thing to do. And then, like in genomics, you can then take all of those data and you parse the data for perturbations in the data set, outliers, perturbations, subgroups of the data, and look at those, and then you ask yourself the question, okay, if my subject sample, whatever, have this attribute, what might that be predicted to cause in the phenotype, and then test that with a phenotypic experiment. All right, so that's a different way of thinking. All right, a few of you have read the paper describing this, so I'll go through it quickly. My colleague, Chuck Venditti, who works on metabolic genetics, had a set of patients who had this rare subset. So this is a subset of a rare disorder. The rare disorder is, let me get this right, methylmalonic acidemia. That affects about 1 in 50,000 children. This is a subset of that phenotype, combined malonic and methylmalonic acidemia, which has the most unfortunate acronym of CMAMA. I didn't come up with that. All right, what is that disorder? Like many organic acidoses, these kids are born and look completely normal at birth. And then as soon as you start giving them protein, they develop organic acidosis, usually infantile onset. They have metabolic decompensation, CNS infarction, coma, and often death. Inherited in an autosomal recessive pattern, as are most metabolic disorders. And so we said, this is straightforward positional cloning, next generation genetics now. Let's sequence a trio, a child and his two unaffected parents, and look for variants that might cause this disease because we had no idea what caused it. Now I want to take a step back here a little bit and talk about the difference between Mendelian genetics and common genetics. I live totally, for reasons you'll see later, in the Mendelian genetics world. That world is part of this graph. So this graph talks about two things. The penetrance of a variant, that is the likelihood that if you have the variant, you manifest the trait, the phenotype, ranges between 0 and 1, and then how common that variant is in the population. So there's an entire spectrum of variation. There's not much up here, but there are rare variants, some of which cause highly penetrant phenotypes. There are common variants that have low penetrance. We live up here in Mendelian genetics because the odds are enormous. If you have the variant, you will have the trait, which makes it much easier to do correlations. So if you're looking for rare variants that have high penetrance, they share some attributes. That attribute is that they tend to lie in exons of genes as opposed to introns or the space between genes. You might remember that genes have promoters, they have exons, they have introns, they have enhancers, and that if you look at the open reading frame, about 80 to 90% of the variants that are known to cause Mendelian disorders lie in the open reading frame of a gene, which is made of a subset of the exons. So if that's true, and you want to find those variants, and you can find that by a shortcut of only looking at exons instead of looking at all the other DNA, especially since exons are only about 1 to 2% of the DNA. So you want to look at exons, you want to sequence exons, that's called exome sequencing. Another word for the general approach is next generation sequencing. What I haven't figured out is what the next round of machines is going to be called. Next gen sequencing, I don't know what they're going to do. But anyway, we call it next gen sequencing for better or worse. Basically, you chop up the DNA into pieces, and some of those pieces contain exons, some of those pieces contain introns, some of those pieces contain intergenic DNA. And then you use an affinity technique to discard the stuff that you don't want to sequence, the stuff that isn't exons. And then this is the real crux of next-gen sequencing right here, which is that you take those little pieces of DNA and put them onto a solid matrix, and you do that a lot. So there's hundreds of millions of pieces of DNA adhered to this glass slide. And what you do is you amplify those in situ, so that little spot, and you basically clone that piece of DNA. So this is one clone. Whoops. There it is. Wrong button. This is one clone of DNA. This is a second fragment. This is a third fragment. And then you sequence them in place on the chip, and you do it one nucleotide at a time for every one of those clusters on the entire slide, of which there are hundreds of millions. And that is conceptually all that what we call is parallelization. We have parallelized the sequencing process so that we do hundreds of millions of sequencing reactions simultaneously and assay them one at a time by taking a picture of the slide, and the color of that spot on the slide tells you what the nucleotide was that's added. So you generate lots of those little sequences. They currently range in the size. Our sequence reads are now about 150 base pairs. And then you feed them into a computer, and the computer aligns those sequences with the reference genome. Big issue there, but we don't have time to do that right now. And you line them up, and they generally pile up over exons, because that's what you selected for. You pile them up, and then you look at them, and you say, Look right here, there's a string of 4 Gs, and all of these reads have Gs in all those positions. The patient must be homozygous G at that position. Here you have a slightly different situation. About half the reads are As and half the reads are Ts. The reference is T, so this person is a heterozygote for AT at that position, and you do that across the genome. You can also do it for whole genome sequencing, what is typically called whole genome sequencing, although it isn't whole. And talk about that later. And look across the entire genome, include the exons, the introns, and the intergenic sequence, and you can get almost everything in the genome. Okay, so you apply that to a kid and 2 parents with some recessive Mendelian disorder, and what do you get? So from that sequence, we got about 50,000 individual variants in those 3 people's DNA when we compare it to the reference sequence. All right, then what the heck do you do with 50,000 variants? You filter. And you start to say, okay, I'm going to filter that for rare variants. I have some estimate of the frequency of my disease. I can make reasonable assumptions about how common that should be. I can also make assumptions about the fact that the kid should have 2 variants and the parents should have 1 each. I can exclude all kinds of variants for that. And I go from 50,000 variants to 12 variants by using these simple filters. Then what do? Well, I look at those genes and I say, what do we know about those 12 genes that fit these criteria? And it turns out that one of them has a predicted mitochondrial leader sequence, even though it's never been associated with any disease before in a human. And I know that most metabolic disorders metabolic reactions take place within the mitochondrion, and I can reasonably guess that that is the one out of the 12 that is my gene. Sequence that in 7 additional patients, and turns out 6 out of the 7 of those patients have 2 rare variants in that gene. Odds of that are infinitesimally small by chance alone. Chuck's group did all the functional data complementation to confirm that that is the gene that is mutated in patients with this disease. Okay, so that's a pretty straightforward find the gene that caused the disease paradigm. Not very novel or interesting. Arguably, that's just hypothesis testing medicine, hypothesis testing research, and so that's the usual drill. But we then turned and did something quite a bit different. We asked a different question, and we said, okay, this is an uncommon subset of a rare disease. How common actually is this disease in the population? Because it's really hard to get estimates from clinics and things like that of how common a disease is because of all the biases in ascertainment. Why don't we look and see how many carriers there are in the population? And I can use genetic principles to calculate, if I know there's so many carriers, how often there will be homozygous or compound heterozygous patients in the population. So what you need is a set of a large number of people who've been sequenced for this gene, for whom you did the sequencing for reasons completely unrelated to this disease. We want 0 ascertainment related to this disease to get an unbiased sample and find out how many carriers there are. So to do that, we used a different cohort of patients that we have under study at NIH called ClinSeq, which is 1,000 people who were, some of whom have atherosclerosis, some of whom don't, but they were consented for complete molecular characterization, including any assay we can apply to them, like sequencing. And we can do potentially any downstream post hoc phenotyping on them that we propose after we have sequenced them. So it's sort of an iterative post hoc phenotyping approach. You can begin to see where this is going. So if I have 950 of these people have been sequenced, I've got 5 trillion base pairs of sequence sitting on this laptop computer, and that includes 2.5 million different variants, huge numbers of variants. And I can begin to look at those and ask questions of those data. So I asked the question, how many of my patients who were ascertained for reasons unrelated to this disease have what looks like mutations in this gene? And this is what the data look like. So you can see, like that graph I showed you with rare and common variants, okay? You can see rare and common variants. So here's the number of participants who have a genotype, what type of variant that is, whether it's non-synonymous, frame shift, or splicing. I don't have any synonymous on here, but we do get lots of synonymous variants. And how many people of those are homozygous reference, heterozygous, and homozygous non-reference? And those of you who have a sharp eye might look at this row and say, what the heck is up with this? Okay, remember, these people were not ascertained with respect to having any attribute of this disease. And they're adults. This is a pediatric disease, right? Okay, but here's one patient sitting in our database who is homozygous non-reference for arginine to tryptophan change in that gene, predicted to be highly deleterious. Two explanations for this. The first explanation, the most likely, is that, oh, and by the way, I forgot to tell you, this arginine 471 tryptophan variant was found in a couple of the children that had that disease. Okay, so what are the potential explanations here? The first one, which is surprisingly, depressingly common, is that I have falsely attributed pathogenicity to this variant, right? Because here's what we do in genetics, and we've been getting away with murder for years on this. Take a group of patients, take 10 patients with some rare disease, you sequence them and you find that 9 out of the 10 of them have a mutation in gene X. I can conclude from those data that mutations in gene X cause that disease. That is a valid conclusion. What is not valid, and we have very conveniently ignored over the years, is that it is not necessarily the case that all 9 of those mutations are actually causative for that disease. Because you know, in one of the 10 patients had the disease and doesn't have the mutation, and that means they probably have the disease for some other reason. And how do you know that that's not true for 1 or more of the people in the 9 group? They actually have the disease for some other reason, and you have coincidentally detected a benign variant in your gene and falsely attributed causality to that variant. And you might be shocked to learn that genetic mutation disease databases, about 5 to 15% of all of the mutations in those databases that are said to cause disease are actually wrong. Really big problem for our field. So, if I'm being self-critical, I would say that's probably what I did here. I have falsely attributed pathogenicity to this variant. But I can actually answer that question by going back to my research subject in this case. I go back to this patient, and it turns out that she is a now 66-year-old lady. In the studies, she has experienced 4 traffic accidents, been in 4 car accidents. Now, how on earth you can get car insurance after you've had 3 traffic accidents in 2 years, I have no idea, but she did. But seriously, she was complaining of significant neurologic problems, increasing memory difficulties, bladder incontinence, which had been unexplained and they couldn't find a cause for, physician. And then we looked at her metabolism and looked at her metabolites. And when we did that, we found out that she had 100 times the upper limit of normal of methylmalonic acid in her plasma. She had 70 in her urine and 11 micromoles of malonic acid in her plasma. Now, these are wildly abnormal metabolic assays. We scanned her in our research scanners, and it turns out she has multiple high-intensity signals compatible with small infarcts that are like small that the kids get in the nursery. All right, so what does all this mean? The mundane conclusion from this is that this is proof of causation, that ACSF3 mutations cause combined malonic and methylmalonic acidemia. It's not the way we normally prove it, right? We prove it normally by starting with people with the disease and showing that they have mutations in the gene. Well, what did we do here? We pulled someone out of the general population. We have sequence data on them for no good reason whatsoever. Picked one of those 1,000 people and said, you have 2 mutations in this gene that you're not supposed to have, and discovered the phenotype in the patient. Okay? That's a different way of proving causation. But I think more importantly is this, is that we completely misunderstood what this disease was. We thought this was a pediatric onset, typical metabolic acidosis with decompensation, stroke, infarct, and death, but it's not. That is but one part of the range of that phenotype. And what it is instead is a metabolic phenotype that has a wide range of expression and severity. And the kids who decompensate in the nursery are just a subset of patients who have this actual disease phenotype. And some people have a pediatric presentation, and some people have an adult presentation. And we're actually starting now to find more adult patients who have this, and it turns out that these are patients who are walking around with what's called atypical multiple sclerosis. What? Multiple sclerosis, completely different disease. Well, it's just that they have a unstable neurologic phenotype that manifests in memory and neurologic problems, and when you test them for the typical attributes that patients with multiple sclerosis have, they're all normal. Okay, so they act like they have multiple sclerosis, but they don't. They actually have this disease. So what it proves to us is that we had no idea what this disease was. And if we had only done hypothesis testing research, we would have never discovered how ignorant we were about this disease. But the genomics told us that the disease was much different than we thought it was. Of course, so then you ask the question, well, why do some have the pediatric, some have the adult? It's probably modifiers, probably environment, and we have to work on that as well. Okay. So the thing about hypothesis testing research is that it does work. You can find generalizable knowledge from that. It's useful. We should continue to do it. But it's actually circular reasoning. Right? We say what we think the disease is, and then we prove what the disease is caused by, and we say that's what the disease is. That's only because that's all we started with in hypothesis testing research. If you do hypothesis generating research with unbiased ascertainment of phenotypes, then you can discover what the true spectrum of the phenotype actually is and understand the range of severity of the disease using iterative research. Okay, so what is this really about? Talked about tools at the beginning. As computer scientist, Alan Kay, one of the smartest people on the planet, he's one of the people who actually came up with what about 15 or 20 of you right now are using object-oriented computer programming. You have on your screen pictures of things, and you are computing by manipulating pictures of things instead of writing command line codes right now. The person who invented that is Alan Kay. It's a radical solution, a radical proposal in the 1970s not to do command line computing. Alan thought that you should do that. And what he said is this, that creativity is a situation where you are in a transition of context. You are started in one context and you are moving into another context. And that's where surprises happen, and surprises are discoveries. And what our job is, he's talking about computer scientists, I would say it's the same for us, is that to remember whatever context you're in, there are other contexts. And that you have to think about the other contexts that may exist and ask hard questions about, is this the only way that we can think about this problem? And he was a radical thinker, and he said, the heck with the command line. I don't need a command line. I want to compute differently. I would say in biomedicine, we have to say you don't just have to test hypotheses. You can generate hypotheses using data first. The disparaging term for that, does anyone know? Study sections love this two-word phrase. Exactly. That's the disparaging word. But the problem is, yes, it is a fishing expedition, and guess what? When you go on fishing expeditions, sometimes you catch fish. I'm okay with that. Okay, so predictive medicine. So that's, I'm talking about the clinical research context. Let's talk about medicine. Do we want to apply this paradigm to medicine? That's not research, it's a practice paradigm. So what would this be? And there's other words for, I think we talked about that earlier, personalized precision medicine. But the notion is you want to apply preventions and treatments to people in a predictive pre-hoc manner before they have disease manifestations. You want to maximize efficacy, minimize side effects, treat diseases before they occur, prevent diseases. Very different from our current paradigm. I don't know how many of you have your eye on internal medicine, probably more than a few of you. Internists hate this lecture. Okay? The internists in general worship at the altar of the differential diagnosis, right? There is a chief complaint. After the chief complaint, there is a physical exam. After the physical exam, there is a differential diagnosis. And then you select the test based on the differential diagnosis. This paradigm works. It's a good paradigm. We should keep doing it. And when a physician is presented by a sick patient, this is going to be, for some time, the best way to approach that problem. That is not to say that it's the only way to approach that problem. Because again, and you're going to, if you're awake, you're going to be thinking this is a little bit repetitive, this lecture. You could do it differently. You could bring the patient in and do your basic intake consent for care, generate data on that patient, and then use those data, look for patterns, perturbations, outliers, etc., predict what disease that is telling you your patient might develop or how you should manage that disease, and then do further evaluation or treatment management to minimize that. Okay, so we're talking about predictions. To do that, you need some assay that broadly assesses that person's probability of having some occult disease, right, a disease that isn't obviously manifest or present, and or future risk or modifier or whatever. Now, this is not new. The best example of this is called the routine history and physical examination for a healthy person. We do routine surveys of patients' risk for occult or future disease. It's called a physical exam. It's a broad survey of risk for occult disease. That's what it's all about. And pediatricians will be used to the concept of newborn screening. We screen patients who have no apparent sign of disease to see if they have an occult disorder like hypothyroidism, even though there's no reason to think they have it. We use those data. So predictions are part of medicine. Using genomics to do predictions are trickier. This is often misattributed to Yogi Berra, but it was actually Niels Bohr, who's not as funny as Yogi Berra, but arguably a bit brighter. And he said, prediction is very difficult, especially if it's about the future. And that's sort of the crux of the problem. It is hard to make predictions in any domain. Of course, these guys were talking about quantum mechanics, arguably a little less complex than medicine, but maybe not. And the guy sitting next to him, equally clever fellow, said this: yes, but occurrences in this domain are beyond the reach of exact prediction because of the variety of factors in operation, not because of any lack of order in nature. That, to me, sounds a lot like genomics. We know about some components of risk. We don't know about others. There's a lot of factors in operation, some of which we can assay, some of which we can't, but we can operate on this system with incomplete knowledge. Okay, all right, so let's keep moving. All right, so incidental findings, predictive medicine. I find that when I give this talk, if I call it incidental findings, people get all annoyed by it because they don't like incidental findings. If I call it predictive medicine, they love it. So call it what you like. But let's take the idea of taking a person out of the general population, sequencing them, finding a susceptibility for a disease, and treating them. Can we do that? We've done it in several ways. We've done it with cancer prediction. And in fact, I would actually urge anyone who reviewed our paper on cancer susceptibility, there was a paper published last week, I think it was in PNAS, by Mary Claire King's group, proposing population-wide screening for hereditary breast and ovarian cancer susceptibility to sort of extend that model. It's a radical suggestion. I think it's ready for research. I don't think it's ready for clinical implementation, but I think it is time to consider that question. I want to talk a little bit about using that same approach for cardiomyopathy. So so we've done this, as I mentioned, for a few things like cancer, cardiac disease, and malignant hyperthermia, all traits that are theoretically predictable by genomics. So we took our 870 people and evaluated them for all variants in 63 genes that are known to cause either cardiomyopathy or dysrhythmia, and set very high thresholds for pathogenicity. Again, we got to avoid that problem of not falsely attributing causation when it doesn't exist. So how do you do that? You set very high thresholds so that your positive predictive value is high and you sacrifice some sensitivity. What did we find? So in the cardiomyopathies, we found 3 variants that we were convinced were pathogenic amongst these nearly 1,000 people. First one was a myosin heavy chain splice variant, and when we went then and did the iterative research, clinical research on the patient, we pulled out their NIH record, and it turned out that this person had, for unrelated reasons, a MRI of the chest. And ironically enough, of course, what other field has had the most experience with incidental findings? Radiology, right? Lots of incidental findings in radiology. They have as much of a problem as we do on that. Turned out this patient had had an MRI, and the radiologist had missed the radiologic incidental finding, because when we looked carefully at it, this person had an anatomic phenomenon called left ventricular non-compaction. Your heart starts out as a loose meshwork of muscles, and in development consolidates into solid muscle with a few clumps of muscles that are connected to the heart valve. This disorder is the failure of that compaction process, and you can see that on MRI. We pulled this person's MRI, and it was obvious that she had left ventricular non-compaction. 2 patients who had cardiac hypertrophy, who both had mutations in the myosin binding protein C3, and both of them on further evaluation had cardiomyopathy. So all 3 of them had abnormal imaging, and interestingly enough, when we look at their family history, what did we see? We didn't see diagnoses of cardiomyopathy. What we saw was unexplained sudden death, okay? So it's hard to know the cause of sudden unexplained death, but one thing we do know is that cardiomyopathies predispose to sudden cardiac death. So if a 24-year-old man has an unexplained death from drowning, what could that be? In fact, it could be that he had cardiomyopathy and died in exercise, because we know that happens all the time. We also looked at the dysrhythmias, found 3 variants that we thought were pathogenic. All of them had subtle abnormalities on their electrocardiograms of dysrhythmia, and all 3 had histories of sudden unexplained cardiac death. All right, so you can take exomes from people without knowing anything about their phenotype, sift them for variants in genes that cause disease, and find variants that have pathogenic consequences in what you thought was a healthy, otherwise healthy person. You do need to use high thresholds, because if you look at all genes that cause cardiomyopathy, the average number of variants per person in our cohort, anyone want to guess what the average number of variants per person is? What do you say, 1 in 10, 1 in 50? It's 1 in 1. There's almost 1,000 variants in our 1,000 patients. And you know that those diseases are nowhere near that frequent. So you know most of those variants are in fact benign. It's a subset of them that are pathogenic, and it's our job to figure out which one it is. So you have to have high thresholds, or else your false positives will go through the roof. But this approach to medicine is criticized because of its poor predictive value. But if you look at our data, what's the only reasonable conclusion you can make? If I pull out from our data 6 people who I think have pathogenic variants, and all 6 of them have the phenotype I predict they ought to have, what mistake am I making? My sensitivity is probably poorer than it should be. If my positive predictive value is 1, my sensitivity is much lower than it should be. That means my criteria for mutations are actually too stringent, and I need to relax them a little bit and find more patients to increase my sensitivity, because if your positive predictive value is 1, you know you have lost sensitivity. Okay. All right, now I want to talk to you about a case study of cancer in predictive medicine, which is this. So we had in this study, it's primarily an atherosclerosis study. One of our probands, 56-year-old person who had a positive family history of early onset atherosclerosis and myocardial infarction, so he had enrolled in the study, and he's clearly an affected individual. He was very interested for us to try and figure out why people in his family were dying of early onset cardiac disease. We take family history for all participants as they come into the study, and it turned out this family history, other than the cardiac disease that I mentioned to you, was notable for him having a sister who was 3 years older than him who had ductal carcinoma about 10 years previously. And for various reasons, and good reasons, even though she was offered genetic testing at the time, she chose to decline genetic testing for breast cancer susceptibility, and her oncologist was comfortable with that. Okay. In our proband, we found this variant in his genome, which is a very well-known 2-base pair deletion in the BRCA1 gene, which is in fact well-known because it is very common in persons of Ashkenazi Jewish descent, of which I think 18% of our participants fall into that group. All right, so what do you do? So here's a 56-year-old guy who came in for heart disease. And we call him into clinic and we say, you know, Mr. Smith, you have a mutation in a gene that causes hereditary breast and ovarian cancer. And his reaction is, no. He is annoyed with us. He is disappointed in the outcome of his participation in our study because we did not find what he wanted us to find, which was the variant that's causing the early onset atherosclerosis in his family. So he is irritated and unhappy with us and dismissive of this result because it doesn't answer the question that he wanted answered. Okay? So you take that as it is. And what's our job then? Well, our job is then to convince him that he's wrong. Now, we're not supposed to do that. We're supposed to be very, geneticists especially, we're this like kind of lovey-dovey, fuzzy, counseling kind of physician, right? And we do whatever the patients want, and we try and make sure that they get exactly what they want from their testing and all that stuff. And we're not doing that here. Saying, no, Mr. Smith, you're wrong. Your opinion about this actually doesn't matter. We are going to tell you what you need to do. And what you need to do is tell your family about this, because you have people in your family at risk for this disease, and they need to know about this. And this took us almost 2 hours to try and convince this guy of this, because again, he didn't want it. Okay, so he then shared this, took it home, and he in fact did what we asked him to do, shared it with his sister, who went back to her oncologist and discussed the fact that her brother had this test from these crazy guys in Bethesda, she lives out of state, who sequenced him for no good reason, found this, and what should she do? And he said, well, given your history, you probably ought to now undergo that testing that you declined 10 years ago. So she tested and was positive for that same mutation that her brother had. All right, so what do you do? Okay. Now, it turns out for her ductal carcinoma 10 years ago, it's really people are so interesting. Her management, she insisted that the management of that ductal unilateral DCIS be treated by bilateral complete mastectomies. Very odd thing to request. But that's what she wanted to do. That's what was done. But she did not have a hysterectomy and oophorectomy for the ovarian cancer risk. So when she came back positive for this variant now, she underwent the hysterectomy and oophorectomy. Now, when that's done, in an individual who has a mutation in one of these two genes, BRCA1 or 2, there's a protocol that the pathologist will undertake, which is serial sections of the fallopian tubes to look for a disease called serous tubal intraepithelial carcinoma, STIC. And it turns out, for those of you who aren't HBOC aficionados, what we call ovarian cancer isn't ovarian. How many people know that? It's not ovarian. Good. It's actually fallopian cancer that metastasizes from the fallopian tube to the ovary, seeds the ovarian epithelium, and then from there metastasizes to the peritoneum. That's the natural history of that disease. We now understand that. And that's the mechanism in people with this mutation. So what you do when you do a prophylactic surgery is you take that those tubes out, and the pathologist does serial sections about I think it's almost every millimeter, and examines every section of that tube to look for occult carcinoma. This is the result in this patient. So here's one section of one of her tubes had this lesion in it, right here. You can see the increased staining on the H&E. It's positive for BRCA1 and positive for P53. This is classic serous tubal intraepithelial carcinoma present in a single section of one of her tubes. And so the protocol then is that patient has to go back to surgery and do two things. They have to do a node exploration to look and see if any nodes are positive, unlikely, but it does happen with isolated STIC, and what's called peritoneal wash, which is you just put in tons of saline into the abdomen, slosh it around, pull it out, centrifuge it, and look for malignant cells. Both of those were negative in her, so she is considered to be cured for the pre-lesion of ovarian cancer based on her screening. So what should you conclude from that? Now, the value, the paradigm in clinical genetics is that patients only undergo testing and evaluation when they come to you and ask for that and have full consent for such testing. Full patient autonomy. That is our paradigm in clinical genetics. We completely violated that. We tested someone without specifically consenting them to genetic testing for HBOC. This man was sequenced, right, for his whole genome, ostensibly for heart disease, but there's other genes in there, like 19,999 other ones. We did those at the same time. Was he consented for that? No. Did he want the information? No. Did he know what to do with it? No. We told him what we thought, how it should be managed, and it led to an outcome that the family is completely happy with. So it really begs the question about our whole paradigm in genetic testing that you can only do it with informed pre-test consent and the autonomy of the individual to decide whether or not they want that test. Now, I'm not saying, based on this, that we should test people against their will. That is ridiculous. Can't do it. But it does beg the question of how you manage patients with respect to what they say they want and what they think they want with what they want. And that's a hard thing to do in medicine. It's going to get harder in genomics. We are making that problem bad, and we're going to have to figure out how to work with that. So predictive medicine has a lot of pluses to it, but it has some minuses too. Those are important to keep in mind. The glass is half full. The glass is half empty, and man, does this evoke furious arguments among my colleagues. Just furious. So is it half full or is it half empty? Now, the half empty people say, you knuckleheads in genomics, you can't interpret most of the variants you find in these genomes and exomes. What do you think you're doing? That is true. We can't interpret nearly all of the mutations that we find. And it is also true, glass half full, some of the variants, we can interpret them, and we do know what they mean. And when they're there, you can use them. Another criticism, glass half empty. You don't know the penetrance. Remember we talked about the penetrance graph? Okay. Here's another bias in medicine that we have that's really a serious issue. That is, we have been studying, over the past 30 years, families with genetic disease. We have biased our studies towards mutations that are highly penetrant because they can only cause familial multiple occurrences of the disease if they're highly penetrant, right? So by definition, we're biased towards the highly penetrant ones. The criticism is, okay, now you start ascertaining people from the general population. These are not loaded families. The penetrance is going to be low, and you have no idea how likely it is the patient's going to have the disease. Probably true, but on the other hand, I did not know what the penetrance of that del185AG mutation was going to be, but you know what? Common sense would tell me that it's very, very unlikely that that range of penetrance in that person includes the baseline population rate. I don't believe that. At some level, that person's risk for HBOC is higher than the population average, and that person can make a decision along with their physician on how to manage that, even if you don't know the exact number. We talk about evidence-based medicine. We have to have evidence for everything we do in medicine, and again, the criticism of us knuckleheads in genomics is there are no studies that show that there is true clinical utility in sequencing individuals. That is true. But you can generalize, and physicians do that every day in every office in the country. They have a situation with a patient that isn't exactly like the beautiful randomized double-blind study in the New England Journal of Medicine. And what do? They generalize from the information they do know and apply that to their individual patient, and they make it work as best they can. That's one of the key things in medicine, is that you have to use the information you can, because you don't have the luxury of saying to the patient, let's wait 15 years until they get that randomized double-blind study done, and then we'll decide what to do. You don't always have that. You rarely have that. So why all the fuss in genetics? The fuss in genetics is because, and we have created this problem ourselves, we have made ourselves into the most exceptional discipline in medicine. We think, I'm talking about we other than the person standing here at the podium, that we are the most unique, most clever, most different physicians there are, and therefore we should have a completely different set of rules for how we practice medicine than do other physicians. And I think that is a very problematic point of view. Truth is, this is just medicine. Sequencing is just another test. We have a test, we have a result, we interpret that result, we use it to change the care of our patient. There's nothing new about that. That the instrument is, really fancy. We use really cool instruments, and they have, glowing blue lights and stuff on them. They're really cool. Cost $750,000 a piece. But what comes out of that machine in the end is just another test result, and we have to use it. So we can use it, and we should go forward with that. What can you conclude from all this? So you can now, because of sequencing technology, you can begin to think about doing hypothesis-generating clinical research. We're doing it. It can work. And you can discover things that would be impossible to discover with hypothesis testing research because you have broken that logical circle, that circular logic of hypothesis testing research, and allowed new conclusions to emanate from the research that you couldn't predict. If you do a phenotype-driven assessment of patients who've been sequenced, you can find average 1 out of 50 people. I take 1 out of 50 people off the street. They will have a mutation in a gene that's predicted to cause a human disease. That's a lot of people. And it's not just zebras. They're not just rare diseases. These traits are all around us, and we have lots of examples. I only showed you a few of them. There's several hundreds of variants in these people that cause human disease. And we look at all null variants across genes. We can find undiagnosed disease, and nearly half of those people have a disease. And again, the critics will say, oh, you knucklehead, you're wrong half the time. No better than a coin toss. Yeah, that's true. But on the other hand, if I find that this patient has a variant that's 50% predictive of some Mendelian disorder, and their prior probability of having that disorder was 1 in 25,000, I have changed that individual's odds of having that disease from 1 in 25,000 to 1 in 2. Now, you can complain about the 1 in 2 all you want, but I'm thinking about the difference between 1 in 25,000 and 1 in 2. And I, as a clinician, I guarantee you most patients think differently about themselves when their odds are 1 in 2 compared to 1 in 25,000. And I can use that information. Now, all that being said, I would not argue today to you that genome and exome sequencing should be applied to healthy people for screening purposes. We are not there yet. It costs too much money to do that for no indication whatsoever. But at the same time, the technology is very useful for patients that have rare or unexplained diseases, and there are many more of them out there than you would like to think. This is not a rare problem. You can do predictive medicine. It's the same thing as incidental findings. So when you do an assay, a genome scale assay on an individual, you should use all the information that's in there that's usable. Not just close your eyes and look at the things that the patient's asking for, because as my case suggested, the patients don't always know what answers they want. Sometimes you have to answer questions that the patient doesn't ask to do what the patient needs you to do. And that's a hard thing to do, but it's part of medicine, and predictive medicine is, in the end, just medicine. So where are we going here? We don't know where we're going. And predictive medicine's hard. Predicting where a field would go is even harder. Go back to Alan Kay, and he said the best way to predict the future is to create it. So can I predict where medicine is going to go? No, but actually we can start to influence where it's going to go. And we can, by the work we do that with sincerity and with integrity. We're not just fooling around. We're trying to change medicine for the better. And it's likely that some of these things that we're doing are going to be, for those of you who are in medical school here, things that you are going to be practicing in 20 years, whether you know it or not. We wrote a review article recently, and I use this as a barometer not to brag, but to make the point that when this technology appears in something like the New England Journal of Medicine, it's here. Like the little girl was a poltergeist. They're here. Okay? So like it or not, the genomicists are here. This is part of medicine, and this is where we're going to take the field. Lots of people helped me with this. Thank you all for coming, and I look forward to any questions you might have.
Thank you, Les. That's spectacular. Chris, question?